So our next speaker is Dr. Bennis. And the title of his talk is Cell Line Screening.
So what I'm going to tell you about is our efforts to understand better how we can harness the molecular characterizations of cancers, and that means cancer genome and other dimensions, to translate that knowledge into at least hypotheses that will lead the development of therapeutics or biological studies that will help the development of therapeutics. So here we are. So I'm directing the Center for Molecular Therapeutics at MGH Cancer Center, and this center was established by Jeff Settleman and Daniel Eber a few years ago. So what I was telling you about is this, is that we try to take all these new characterization of the cancer makings, the molecular making of cancer, which a lot of is known in terms of cancer genome and a little bit less about other types of dimensions, so that proteomics and other types of characterization like that. And we try to understand how we can functionalize this, make it so that we understand the undergoing responses to drug, so that we can extract biomarkers, we can extract other information that will be useful for drug development or drug application. And in fact, drug application is probably where this type of approaches has been the most useful so far. So the reason for that is obviously very clear to everybody in the room. Probably that's because we know that if we identify a specific genetic lesion for which we have the correct drug, we can get pretty good responses in the clinic. And so the canonical example is obviously BCR-ABL translocation in CML and dramatic responses to ABL inhibitors. And of course, new therapy are coming up that are the same kind of relationship between a given gene and a drug, such as EGFR mutation in non-small cell lung cancer and various inhibitors of the kinase with several new type of new generation of inhibitors that are better than these ones, for example, and a whole slew of other similar therapeutics. So I put these slides here because I think it's important to understand that although we want to identify pathways and we want to put things into pathways, equivalent activation of a given pathway to something that will be leveraging or leverageable rather for therapy, there's actually a lot of subtleties to the game. And by that I mean that this is actually exemplified by this particular study very well, is that if you look at responses of non-small cell lung cancer patients that have an EGFR mutation versus wild type to gefitinib, it actually works really well. It works better than the standard of care at the time. And so, you know, this is a good identification of a specific genomic variant that you can act upon using an inhibitor of directly of that particular genetic variant of the kinase. In fact, the inhibitor inhibits EGFR mutant or EGFR wild type equivalently in this case. However, if you look at patients that have high copy number, so seemingly something that should also activate the pathway, they are not responsive to the therapy as much as the mutant, and in fact, they're undistinguishable from low copy number variants. So there is a need for fine granularity, and I think I will try to give you a sense for that we can capture some of that, or at least something that might be related to that in this large-scale cell line screen that I will describe. So how do we do what I said about capturing cancer genome and translating cancer genome? So we do something fairly simple conceptually. We take a very large cell line collection. These are tumor-derived cell lines, which means essentially that these are cell lines that are coming from repositories, ATCC, equivalent of ATCC in Europe and Japan, for example. And we treat them with drugs, and we focus heavily on drugs that are either in the clinic or very close to be in the clinic. So we don't do very large cell collections or very large collection of chemicals. We do large number of cells and relatively small number of chemicals. So it's a little bit perpendicular to your typical high-throughput drug screen. And the number is important, and that's obvious by the fact that some of the events that we want to capture are relatively low frequency events in terms of genomes. That means that only 10% of non-small cell lung cancer are mutated in EGFR. So if you have a very small number of non-small cell lung cancer cell lines, the likelihood that you will capture that is not very good. So power is really in the numbers here. And for reference, I've put here the numbers of the NCI 60 panel versus the numbers that we have in our panel. And this is not to say that the NCI 60 panel was not useful and hasn't yielded a lot of important information. It's simply to say that you cannot capture the variations in tumor genomes that we now are aware are out there in the real world if you use a small number of cell lines. So this is a bit of a zoom in of what we have in the collection, and you can tell that for some tissues, some disease subtype, we have a lot more cell lines than for others, and that's due to the fact that simply there are different numbers of cases, but there's also some cancers that just don't have a lot of cell lines because they don't take. So we have some efforts into trying to get this collection more representative in terms of the case number. But so far, this is what we have with very large number of non-small cell lung cancer, some good number in melanoma and breast, et cetera, et cetera. And you can tell, for example, that we have a very low number of prostate cell lines. And in fact, if you look carefully, there's probably about 4 or 5 prostate cancer cell lines that are clearly different in terms of origin, patient of origin. So just to give you a little bit of a dive down into how we're really doing this, we simply look at cell viability, the proxy being either a metabolic stain, which is a resazurine-based staining, or in the past, we don't do that anymore, but we used to do a lot of fix and stain, which means that we were fixing cells and then staining ribonucleic acid, DNA, and others using fluorescent Cyto60 red stain. So basically, that tells you more or less how many cells are there. It doesn't tell you if they died by apoptosis or something else happened, but this is very cheap and it's very fast. So you get a lot of data. This is a small piece of our data. You can't read that. That's purposefully. And that's just to tell you that when you get a lot of data like that, of course, there's a lot of things you can do with it, and there's a lot of things you can get drawn into. So first of all, I wanted to tell you something about the obvious. We want to know whether we can do this in high throughput and whether we can capture what we should capture, the most obvious things that we should capture. So if you look at this, this is simply showing you the response of imatinib, a BCR-ABL inhibitor, across the whole cell line collection with some classification by basically primary site, tissue of origin. And as one might hope, what we get is that we get, and this is a relative IC50, in other words, it's centered so that the total number of cell line or the total repartition will be centered around 0 in terms of IC50. And it tells you that, relatively speaking, CML lines are extremely sensitive to imatinib compared to everything else. This is actually not a trivial result. And that's not because we didn't know it was going to happen, and that's not because we're reinventing anything specific here. But what's interesting is imatinib is not a very clean drug. So we talked a lot about dirty drug and clean drug and how you can act better with a dirty drug or a clean drug. But this is not a very easy definition, nor in this system or in terms of clinic, because imatinib inhibits a lot of other kinases at equivalent potency, yet we get a phenomenal selectivity for CML. And of course, the obvious reason, or at least one potential obvious reason, is that all the other cell lines don't really care about the other kinases that are inhibited by imatinib. They don't care about ABL, but they also don't care about PDGF receptor in the context of the assay. They don't care about all these kinases that potentially could be deleterious to this statistical association. We get these phenomenal results. So potentially in patients, you could say that sometimes you're going to have also some compounds that are not very clean, but it doesn't really matter as long as the other kinases that they target don't lead to high toxicity or differential problems of proliferation on other type of compartment than the cancer. Okay, so we can look a little bit more carefully at what we can do with this type of data, and I will start with a small section of what I mean by disease subtype. So we've talked a lot, or we've heard about breast cancer and different types of breast cancer and signaling and how you can slice and dice it and try to understand how network knowledge about the underlying biology could be leveraged using new drugs. And so one of the things that we can do here is we can take several types of inhibitors that are targeting canonical pathway that we know are important for a given type of cancer, such as, for example, TOR PI3K pathway inhibitors or MEK inhibitors and RAF-MEK inhibitors. And we can see whether or not we can distinguish different behaviors, different phenotypic behaviors in the cell collection. And so this is a collection of breast cancer cell lines. And these two classes of inhibitors are clustering. Essentially, the two clusters here are two types of responses that we see across the cell line collection of these breast cancer cell lines. And you can tell that if you look at the mutational status, they actually, this screen here is very good at distinguishing which cells are mutated in PIK3CA. But on the other hand, the HER2 status does not track as well with the different inhibitors. So what it tells you is that you can start seeing relatively subtle phenotypes using enough cell lines to tell you what's going on. So the hope is to use this kind of information to then zero in on underlying mechanisms that will tell you which cell lines that have an HER2 mutation will be responsive to what class of inhibitor using other genetic events. So we'll go back and see what kind of tools we can use to drive directly onto the genetic that is underlying these responses. So now if you look at the gene-centric analysis, in this case, we have 70 cancer genes in this particular analysis. We are looking at 70 cancer genes that are the most commonly mutated in cancers. And so we try again to see whether we can see the main signals that we should be able to see when we do this kind of studies. So we look at 135 inhibitors, lots of kinase inhibitors, lots of other inhibitors, and we're looking at what are the best association between BRAF status, BRAF mutational status, and response to a drug. And so in these plots, what you have is the significance on the y-axis, and then the x-axis tells you whether this event, this genetic event, is associated with sensitivity to a drug or resistance to a drug. And each dot corresponds to a given gene-drug association. So in this case, for example, you see that BRAF mutation is associated, as you might expect, with sensitization to RAF and MEK inhibitor. But lots of other inhibitors that we've screened are neutral in terms of BRAF mutation. So clearly, you can do this in high-throughput. You can have melanoma cells in there. You can have lots of different cells. And what you get is you get what you want out of that kind of approach, which is that BRAF mutation is strongly correlated with response to inhibitor of the RAF-MAP kinase pathway. So we can see a number of interesting biology like that we can capture very well with the screen. Some of them are very well described, although not necessarily acted upon in terms of the clinic, such as, for example, the relationship between P16 that has been a little bit scrambled there, and PRB, an inhibitor of the cell cycle, in this case, CDK4/6 inhibitor, where you get the expected relationship between mutation and sensitivity to a given inhibitor with the same inhibitor here as you might expect from the pathway topology, giving you resistance in one case for RB and sensitivity in the other case for P16 mutation. And the same is true for nuclein, where nuclein is an inhibitor of the interaction between MDM2 and P53. P53 is degraded upon ubiquitination, and MDA2 is responsible for that. So when you have nuclein, you get stabilization of P53, and you might expect that then if you're wild-type P53, you're going to be able to respond, and if you're mutant P53, you won't be able to respond. So that's exactly what we see. What's interesting, though, is that in the field of cancer, a lot of studies have shown that P53 pathway is actually supposedly, at least in cell lines, and particularly in cell lines, inhibited almost all the time, right? That not only you have inhibition of the P53 pathway when you have P53 mutation, but you also have inhibition due to other events when P53 is wild type. And what it tells us somewhat surprisingly here is that when you look at nuclein response, you can explain almost all the responses, although they have some outlier there. You can explain almost all the responses just looking at p53 mutational status. We don't really understand what that means, but what that means potentially is that even though you have other ways of inhibiting the pathway, only the wild type are really responding, meaning none of the, only the wild type are responding, meaning none of the wild type, or not a large proportion of the wild type, are actually inhibited downstream. If you reactivate p53, almost all the wild type are actually acting upon the proliferation. This is a curious result also because when we look at all the other drugs that we've screened, this result is actually not very common. In other words, p53 is not found as a strong correlate of all the other drugs that we found, that we've screened. And a lot of these drugs are targeting events that are clearly linked to p53 activity. So they wire down towards p53, such as the canonical signaling pathways, so PI3 kinase and MAP kinase, etc., are known to wire down to p53. Yet we don't find a lot of p53 correlates in the data set. And so one of the best idea we have to explain that is that what happens is that in a lot of cases, p53 mutation are only part of the story, and the p53 wild type are actually not very good at responding because they are disconnected from the upstream signals. So if you're p53 wild type, you're p53 inactivated not because your downstream signaling is messed up, but because you cannot talk to p53 from the upstream point of view. So far I've shown you a lot of stuff that you already knew. So hopefully I'm going to show you a few things that you don't quite know as well. So we see a very large number of novel drug-gene interactions, some of which we have started pursuing more mechanistically and some of which that are there for anyone to pursue. I'll show you at the end. We have a website for which you can look at, on which you can look at all the results we have and figure out whether it's useful for your own research. And so some of these results are, for example, that are pretty strong statistically speaking, considering the number of cell lines that we have in the collection. For example, mutational event in FBXW7 are related to MS275 sensitivity, or TET2 related to WEE1 CHEK1 inhibitors. So if we look a little bit more about what that could mean in terms of a specific disease, here are represented the activity of MS275, in a specific subtype of disease. So this is a TLL cell line panel. And when we're looking at here is whether, first of all, what kind of strength of effect we have. So MS275 relative IC50 again in FBXW7 wild type or mutant lines. And so you can tell that there is this statistical significance that you showed before represents a relatively important shift in the potency of the inhibitor in the wild type versus the mutant lines. And then if you drill down, and this is only in TLL, so the slide before was showing you across all the diseases. And if you drill down a little bit further, knowing that TLL are often mutated not only in FBXW7 but also in Notch, and in fact most of the mutation in FBXW7 are found in the context of Notch mutation, you can tell that looking at this panel that the double mutant seem to be even more sensitive to HDAC inhibitors, to this particular HDAC inhibitor, I should say, than the double wild type cells. So we don't really know what's going on with that, and so we are looking into the underlying mechanism of that. So we've also screened a bunch of classic chemotherapeutics or cytotoxic drugs, and so the hope was to maybe able to finally get some answers in at least some diseases or some subtypes of diseases for the reason for this variation in response you see in the clinic, maybe get a little bit of a leverage for what genes could be related to a differential response to cytotoxics. And what this graph shows you is that, in fact, when you look at the correlates in the genome, at least in the 70 cancer genes, the most mutated cancer genes that we've looked at so far, and we're looking at a lot more data now, you don't see a lot of strong correlates. So none of the most mutated cancer genes, as you might have expected, but I think it's worth noting that that means we can't really leverage these most common events so far with these classic chemotherapeutics. None of these events are strongly correlated to response to the cytotoxics. And this is exemplified here by the fact that these are the type of p-value we're getting for these 13 compounds, and this is the kind of association we see for the rest of the collection. So you have many more strong correlates in the other drugs compared to the cytotoxic drugs. So this is the overview of what we have. So as you can tell, there's a lot of data points here, and each of these data points, again, is a gene-drug association. And so we have lots of things that we know should be found and that we found very efficiently. We capture very efficiently all these associations that have been leveraged in the clinic so far. And these go from CML to melanoma to a different type of sensitivity for which there is not a lot of understanding so far, and it's not necessarily leveraged in the clinic, so potentially can be useful when you have specific data that you can put this together with. And then most of the thing that you see is that the very top associations, and maybe that's because what we know in terms of the genome is also what we target the most with the drug that we've tested so far, so it's some sort of co-evolution of knowledge going on here. Most of the big associations are pretty much the obvious association that we should find. So not a lot of the very single gene, strong drug, single gene drug interactions that we found. Not a lot of very strong oncogenic addiction type of relationship that we discover with these 130 cancer therapeutics, which again, I should say, target a relatively small target space, right? They tend to target a lot of the things that we've already learned a lot along the years about. But we did find one association that was very strong and unexpected, and this was the association between olaparib, PARP inhibitor, and the EWS-FLI1 translocation that is found in Ewing sarcomas. Oh, that's a little bit of a scramble, but this is okay because this is really a visual. So on the left is a viability assay with a much longer term viability. So I told you what we did most of the time is looking at 72 hours response. So you could say for PARP inhibitor, is that really relevant, and what is the biology behind that? And can you show in longer term assay that you actually have differential activity of some of these drugs? So this is what we did here with a much longer assay, which is more of a cold information type of assays. And so depending on the cell line, this is a 10 to 20 day time frame. And the blue bars that are labeled with this, obviously scrambled label are the EWS-FLI1 translocated lines. And we have a number of sarcomas here and other types of cancers. And what you can see is that in response to PARP inhibitor, we have a very strong response in the Ewing sarcoma. This is a concentration response dependency. And in this type of cancers versus the rest of the collection, even in the similar, in terms of etiology at least that we can understand, type of tumors or type of tumor-derived cell lines. And this was meant to show you that as a single agent, we obtain apoptotic response, again, specifically in the Ewing sarcoma cell lines. All right, so this is one of the associations we saw. And we learn a lot more than that when we look at that kind of very large data set. And one of the things that we've learned is that obviously we weren't particularly naive getting into this exercise thinking that we were going to find a lot of single gene-drug interaction that will explain the whole drug response. But you have to keep in mind that obviously the advantage of that is that if you find a very strong single gene-drug interactions, then you can action that into the clinic. It's a lot more difficult to look for 20 genes correlating with a response and try to go into patients and figure out how you're going to have a patient selection strategy if you have a lot more than one or two genes. That being said, we obviously are aware of the fact that there is a large variation in treatment response in the clinic. And I don't think this is the slide that I was supposed to have, but that's okay. So the idea here is obviously that we know that even if you look at pre-selected patients, and you look at one of the most successful current therapy in terms of clinical responses, vemurafenib in melanoma V600E mutant, there is a large variation in the response range that you see in the clinic, and also a large variation in terms of duration of response. So potentially, we thought that it could be captured in part by the cell collection. And of course, what we see in the clinic doesn't have to be related directly to what we see in cell collection. But what's interesting is that we do see that for this type of agent, as well as for any type of agent, any agent that we've actually screened in the cell collection, there is a large variation in response when you look at pre-selected or when you look at selected cell lines versus wild type. So what I mean by that in this case, you're looking at RAF mutant versus wild type across RAF and MEK inhibitor. And in each case, there's a large difference between mutant and wild type, clearly giving you this volcano plot that I showed you before, this obvious relationship between the RAF, MEK inhibitors and response to in the mutant versus the wild type. Not a surprise. However, you see that there is, for each of them, there's a large variation in response to the inhibitor in the mutant group. And this is not simply due to the fact that for one given inhibitor, you respond, one cell line responds, and then another inhibitor, another cell line responds, and you get this sort of scattering. Because if you look at a clustering of these different cell lines across the inhibitor of the pathway, you actually realize that the same cell lines are responsive to a given inhibitor, and the same cell lines are non-responsive to all the inhibitors. So all the inhibitors are working in some cell lines, and all inhibitors are essentially not working very well in other cell lines. So this is only not even a problem of the type of mutation, because we're looking here only at BRAF V600E mutant lines. Okay? So clearly, we don't understand what's going on here very well, because we have the best selection we can. We have some of these inhibitors are even somewhat better, although marginally better, at targeting the mutant than the wild type. And these are pretty specific, and we don't get very homogeneous responses. What you can see also, and this is borderline statistically significant, is that similar to what we see in the clinic, although not necessarily for the same reason, we do see that melanoma lines are actually more responsive than other type of cell lines, or cell lines that are coming from other type of cancers or of origin. Again, all of these lines are V600E mutant lines. So this is an example to tell you that you need to obviously dig a little further and try to harness the complexity of the tumor, the complexity of the genome, to try to explain some of this variation. So we've done a variety of things to try that, and one of the things that works best, although it doesn't mean that it works perfectly, is a multivariate approaches that basically takes all the information we had at the time, and we have now a lot more information in terms of mutation, but at this point, this was 74 mutational and translocation event, copy number across 459 genes, and these genes are chosen to be the ones that are known, the best known in terms of their involvement in cancer, and expression data across 15,000 genes. And then this data is essentially available in this particular exercise, across 500 cell lines. And we use a linear regression approach to derive essentially predictive models that will tell you which cell line should respond. And then you have in these models a list of genes that might or might not be inspiring. So one of the things that we see first is that a lot of these responses, a lot of this signature of response, or this signature of sensitivity, I should say, of correlates of sensitivity that we extract from these approaches, are actually containing a lot of expression variable. In other words, that we certainly have strong influences of mutation, but high at the top of this list that have strong effect, we see strong influences of expression features. And that couldn't just be because we're covering this particular feature with expression and we're not covering it with mutation, or there are no mutation in that particular feature. So it's not necessarily an intrinsic property of expression versus mutation. It's simply that the biology that we can describe for these responses clearly involves strong influences of expression changes. So this is an example of the drug lapatinib, an HER2 inhibitor that inhibits also other kinases, and showing you that certainly HER2 amplification is important for response. So HER2 amplification is designated by these black squares. And then you have other, such as HER2 expression or MMP28 expression, a metalloprotease expression. This is at the RNA level that are strongly correlated with sensitivity. You might note that these are relatively complex responses, complex model responses, I should say. And that, I mean to say that you see that these lines that are HER2 amplified are actually not the same one that are participating in the model in giving you these complex responses, because the MMP28 expression are actually found in different cell lines. Each of these columns is a cell line. So we can do this for some of the drugs. Sometimes it doesn't work at all. We have not a very strong understanding for why we can do that. Probably we just don't have the right way of measuring the biology for some of these drugs, or we don't have the right way of measuring genome changes or expression changes that will give us the best credit for these drugs. But you can tell that sometimes it works very well. And again, there are sometimes two groups of cell lines, some cell lines that are represented by some of the features in the model and others by different features in the model. So to try to understand whether we're getting anything valuable out of this, we looked at things that we understand seemingly better, for example, MEK inhibitors. And so this is another model of multivariate responses of one of the MEK inhibitor. And you can see that being melanoma, as you might expect, is important to respond. This is kind of what I was saying when I was showing the V600E mutant that have a differential response in melanoma versus other types of cancer. And this is not a complete overlap between being melanoma and being mutant in BRAF, because BRAF mutation is also in the model, as well as other markers of being melanoma. So if we look now at multiple models of response to MEK1 and RAF inhibitors, what we can see is there is a large overlap in the top features, in the features that are represented with the biggest size effect, the stronger effect essentially on sensitivity. And what this depicts is the fact that, for example, as you might expect, BRAF mutation is found in all the model, but a number of other features of these models are shared by these model responses to the inhibitors. And interestingly, several of these features are known MAP kinase pathway regulators. So, that makes us feel pretty good about the fact that we are extracting meaningful biology out of this exercise. So you have both MAP kinase response pathway as well as melanoma response. And again, this is not necessarily a type of modeling that will exclude these if you had melanoma in there, but we think there is actually something going on between the level of differentiation and the responsiveness, which is why you have expression features of the melanoma lineage that are present in these models. So clearly, we can't answer everything with these kind of approaches, and so we're also trying to get our toolbox a little bit more furnished. So we know there are many diseases that we don't target very well, essentially because we don't have models. So we're trying to get more cell lines into the collections, and we're trying to get better variation of genome captures. And so that we will be able to do a lot better now that we know what's out there in terms of genome variation, since there has been a lot of efforts, both in terms of the cell line collection characterization and in terms of, as you know, primary tumor characterization. So hopefully we'll be able to see what we're missing, the major event that we're missing, and try to gather them in the collection. We know that we can potentially address some of the issue or help address some of the issue with limited efficacy, and that we do this by looking at screening cell lines that we've rendered resistance in the lab that are coming from patients that have acquired resistance. We're also looking, as I showed you, at the variation in response that we can call, in this case, de novo resistance. And then there are problems of tumor heterogeneity, and for that, we're trying to get ourselves into a more complex assay so that we can look at single-cell type of response and address a little bit more whether some of the incomplete responses we see are actually due to some heterogeneity in the cell population. And overall, with these models that we have and the overlap between the different models of response we have, one of the things that we can try to address a little better is whether we can in fact discern that some of these private events can be put into pathways using this type of genomic, pharmacogenomic exercise. So, take-home message are fairly clear at this point. We think that this approach can capture biologically relevant information, potentially clinically relevant or clinically insightful information. We don't find, using the drugs that we have now, targeting the space that we have now, a lot of strong oncogene addictions. So that means things are more complicated than one could wish for, but that's not particularly surprising. And we do see that context does matter, and that means essentially that even within a given disease subtype, you're going to have variation, and across disease subtype, you're going to have variations. So in a sense, that's somewhat of an unfortunate finding because you could expect that a given mutation will be very good at predicting response across disease, and I think the pendulum has to go back a little bit in the middle from the completely histological perspective to the completely gene-centric perspective, and that you're going to have to combine the two to really get the right approach. So this is a large-scale effort, so there's a lot of people involved. This is the team at MGH, the current team at MGH, with critical collaboration with Sridhar Ramaswamy Lab for modeling of the data, with Elena Indelman, a postdoc in his lab. We've done a lot of work along the years with my old friend Jeff Engelman, who's always a good advisor in terms of this kind of work. Daniel is instrumental in telling us what to try to do or not. And then we have Adam, who's working with Jeff and I on specific combination against acquired resistance. Adam Friedman is actually in David Fisher's lab, and we are putting together some large-scale combination. And this is the technical team who is instrumental to have this happen. Funding is coming from the Wellcome Trust. This is a large collaboration with a team at the Wellcome Trust Sanger Institute. And we also have funding from the NIH Common Fund for work that I haven't had time to talk to you about that looks at more mechanistic aspects of the response. And this is the full team listing with Mike Stratton, Alton McDermott, and Matthew Garnett that are the leaders at the Wellcome Trust Sanger Institute site. We have a website. Please go there and tell us what does and does not work and what you think is useful and is not useful. There is a lot more data that will come online on this site along the years. We have released more data than what was published in our recent publication in March. You can browse the data in various ways. You can interrogate the data in various ways. You can download all the data on your computer and play with it. So this is it, and I'm happy to take any questions.
Any questions for Dr. Bennis? I was just curious about how you would approach combinations, which we're using, of course, in the clinic, and the complexities of combination indices and combining different doses of drugs, pretty complex.
Sure. So, the combination question is something that we've carefully got ourselves into, and so there's been a lot of work, in the past in terms of conceptual, approaches to combination of therapies. Obviously, not a lot of work has been done across large cell line collections. So there might be specific challenges there that we need to be, aware. So one of the things that we've started doing more intensively is looking at acquired resistant context. And that's because we have a little bit of a better sense of what we might want to bang on. And that is coming a lot from what Jeff Engelman has been doing, and which means that we basically keep one of the inhibitor on, which is the inhibitor that was originally effective and is not effective anymore, and then we put something else together with that. So that's a conceptually, covering a specific space, but, we think we have a relatively good handle in terms of analyzing that kind of data. And we learn by doing that what works and what doesn't work, and we learn the complexity of that. And then we have an effort that I mentioned in passing that we're going to establish with an ultra high throughput system where we're going to go to 1536 well plate and imaging so that we can really go a little bit more open-ended in terms of combination. And so we'll try in the vicinity of 20, 25 drugs against 200 drugs, some sort of rectangular type of approach. And here, we're erring on the side of dense data so that we can actually learn something from the negative data by opposition to going to sparse data and having maybe a few low-hanging fruits and then 99% of obscurity, which means we will take time and we will build a data set that hopefully will be useful not only for us, but also for people to not do it again because we haven't done it with enough density.
Thank you.
Sure.
Questions?
Cyril, one approach to try to understand the variability would be to use shRNAs to knock out specific genes and see if it sort of evens the playing field for
Right.
an inhibitor.
Sure.
Have you tried that?
Yeah, so that's a good and, as you might expect, fun and complicated question. So we've tried that. So one of the idea was, you could say, okay, so what do we do with these lists? This is fine, you get lists, and I have no understanding of what you just told me, and I don't really care because you haven't functionalized any of these lists. So the obvious, potential is to just say, okay, we're going to cross a very different type of approach, which is, available genome-wide siRNA type of screen together with the pharmacology approach. So we started getting ourselves into thinking that we will be able to understand from the combination of the siRNA with the drug by the shape of the response that you have with the SI versus without the SI for a given target that is in the pathway, that we will understand what's going on. So for example, we said, okay, we're going to take the MAP kinase pathway correlates, okay, that we have in these, and we know some of these are in fact in the pathway, so maybe some of these are in the pathway and they haven't been discovered yet, so they are interesting, right? So we did that, and we started knocking down MEK1, 2, and RAF, and adding the RAF inhibitor and the MEK inhibitor. And we don't understand anything. It's a beautiful mess. So I think there are two problems with this. One is the obvious problem of off-target is completely different. There's a huge amount of off-target with SI that, obviously there's a huge power of SI, but there's also a huge problem of SI off-target. And so we're getting better SI, super potent SI that helps us do that. And in fact, it gets rid of a lot of the noise. And the other obvious thing is that the timing of inhibition, the whole way you're actually perturbing the system is completely different when you do an SI and when you do a drug. That's not even talking about the polypharmacology or of dirtiness of the drug, if you want to call it by a more fancy term, polypharmacology, that you will obviously get differential response. But that's simply to say that if you inhibit asynchronous cells rapidly in the pathway with a drug, which is an immediate action, versus when you inhibit a ramp-up effect when you mostly will have with this siRNA activity, you're just not going to have the same result. So that was a complicated answer to the question that we've tried that, and we will continue to try that. But I think it's going to require, similar to what we've done so far, it's going to require a lot of dense data to really be able to leverage things. And we might have a few low-hanging fruits where we'll be very happy to have in a given setup, a given cell line panel, something that works really well, EGFR with RAF or something like that, but I think it's going to be relatively rare. Thank you very much.
Thank you very much.